My name is Nitesh Kumar Sharma, and I'm a postdoctoral researcher at the Mangool Lab, University of Southern California. Today, I am excited to present our work. In this presentation, I will be discussing the development of MetaViz, a tool which is designed to create realistic and diverse benchmark for metagenomic research. Thank you for being here today. I would also like to extend my gratitude to organizer and the chair of this session. I look forward to sharing our finding and discussing metaverse with you guys. Okay, so benchmarks are basically crucial for evaluating the performance of current and future bioinformatics tool, yet often rely on simulated dataset. Even the state of art benchmark CAMI relied on simulated dataset. So our approach is basically aimed to address the limitation of both experimental and simulated dataset by combining the strength of both kind of approaches. So currently there are two types of approaches. So first one is experimental benchmark, where data is generated in wet lab, and which is basically like biologically more realistic, and also you can say like gold standard. But this is kind of like not scalable, and also like kind of time consuming. And second type of benchmark basically generated using simulated based data. In this data set, like this is cost effective and also reproducible, but again this cannot capture the like biological complexity like simulation biases and all. So uh, our solution is basically like creating novel benchmark like through like computational modification. So we are going to use mock community or in vitro mixture read data with like genomes, and ultimately metabase is going to return like a modified benchmark. And as you can see, like it is going to take the like genomes of microbes and in vitro mixtures, and ultimately will return like a different modified benchmark, like multiple benchmarks. So pipeline, like how it is exactly going to return. So like this is the pipeline. As I said in last slide, like it is going to take the genomes of the microbes, and then it is going to map the read onto the genome. After this, it selects the basically like uniquely mapped read and for each species, and then it is going to make the changes into those reads. Like first change it can do is basically like change the relative abundance, and by changing I mean it can like decrease or increase it it can increase or decrease it relatively. Relatively is basically like important here, because we are not going to generate any simulated like new reads. So if we are like decreasing one species read, so automatically like the like second or third species reads are basically like relatively increased to that species, right? So in this way basically we are going to like change these benchmarks, right? So here as you can see, like for species A we have removed some of the reads, right? And similarly this species D read is basically like completely absent from this new modified benchmark. So remove like any species specific read, right? So what are other features of MetaViz? So other features of MetaViz which include basically converting pack bio reads to like mock community, like pack bio reads of mock community into like short reads by reducing the read length, or generating overlapping reads, right? And similarly in nanopore reads, MetaViz is basically going to reduce the read length as well as the error rate in those reads, and ultimately is going to like create the like new benchmark. Okay, so we are going to use like right now we are using these four data sets. Like first data set is 9 strains of HIV, and in this also like 2 strains are also present for HIV. So there are two like data sets. In data set 2, 11 strains of bacterial species are present. Then data set 3, 26 strains of bacteria and archaea are present, and in data set 4, 12 strains of bacterial strains are present. For data set 4 basically like this is kind of unique because it is like pack bio and nanopore data, right? So for data set 1, like what kind of changes we have done. So first, when we mapped these like 9 strains of HIV, we found like 18% read were uniquely mapped. So these kind of changes like MetaViz does with this data, like original data set was like this kind of frequency was there, then we changed it to like, as you can see, like multiple kind of changes are there. Like initially it was like high. Then in this, like it was almost equal, because we have reduced the number of in this first species, right? First strain. And in third modified data, we have completely removed it, right? And also, like we decreased the number for the second strain, so ultimately it came like in scale of these other species, right? So then in second, like the second subset of the data set one, there were like not much of the uniquely mapped reads. So this is kind of, you can say, the limitation of our tool. Like if you we are not getting the uniquely mapped reads, so we are like we cannot change much in the like benchmark data set. Then in data set 2, there were 11 strains of bacterial species, and we found approximately 59.45% uniquely mapped reads. So as you can see, like some of the species were completely removed, and how we exactly changed the relative abundance in modified benchmark data, modified benchmark 1, 2, and 3, right? Then for data set third, so in this we basically like did kind of mix up things. So for data set third, we just like for demonstration purpose, like there were 26 strains were there, but we selected only 5 species or 5 strains for data set 3, and we are basically mixing uh, these data set with the data set 2. And as you can see, again, like there are multiple modified benchmarks like we were able to generate. So yeah, in this also, like we have removed like some of the strains. Like here this was removed, the like this was removed, in this was removed, so like this. For data set 4, where like nanopore and PacBio reads were present, so like for PacBio, because there are like error rates are like very less, so we basically like directly convert these read length to like short reads, right? And for this nanobore, first we map those read to like original the species, and then we reduce the error rate and also read length. So ultimately, using PacBio read, we were able to generate like approximately 40 million single length short reads of 100 base pair length, and from Nanopo we were able to generate like 60 million single length short reads. So scope of expansion for future. So we are going to include more diverse environmental sample for this work, and also we are going to expand the application of this work into immunogenomics, because similar kind of problem exists there also. And collaborate with broader scientific community to like refine the benchmarking framework. Limitation in this work, like first limitation was limited availability of metagenomics mock community. As you can see, like we have used like only 4 data set till now, still we are searching for the new one. And second is basically like the like studies basically does not report the actual reference genome, or like the actual genome they have used, or like version of the genome I'm talking about, right? So in that case, like we have to use the latest version only. So yeah, so that's the also the kind of limitation. So yeah, MetaViz is basically we are developing in Python and Unix framework, and it will be ultimately available on GitHub. So this is our team. I would like to thank to like Dr. Sergey, and also our team like lab members. Thank you. Yeah. So I'm open
Thank you
for
so
any.
much.
Yeah.